Great.
Perfect. So hi again, everyone. Thank you so much for joining us for April Community Call. Today, our speaker is Dr. Elena Rosca. She has been, as I said, a longtime contributor for Reclon. She's leading our hub in Africa. And so she is a professor at Ashesi University, serving as the head of the engineering department. And before relocating to Ghana, she taught at the University of Hull and conducted research in oncoimmunomodulation and the application of focused ultrasound. She has a bachelor in mathematics and microbiology, a master's in microbiology, and a PhD in bioengineering from the Arizona State University. And we're very excited to hear about the work she's been doing in West Africa, the work she's been doing for Recon, and other amazing initiatives in open science and biotechnology. So without further ado, it's with you, Elena. Thank you so much for joining us.
Thank you so much for the introduction, and thank you all for the opportunity to be here and present our work and see what sort of collaborations can we start across the world. Feel free to interrupt if you have questions, and then obviously at the end we can discuss some more and go through other things perhaps that were unclear. So for people who don't know Ashesi University, I thought I would just start introducing the university just a little bit. We are a fairly unique kind of university. We started very small about 20 years ago with 30 students, 4 faculty members, and 4 administrators. And in one of the houses in town, it was a rented property where we actually started. And we are currently on the top of the hill with this incredible view. This is our campus, and we are extending quite a bit to the back of it, where we currently have over 1,500 students. We have graduates and undergraduates currently. Our students tend to do very well in the industry and get jobs right away. We have really a parity between students on scholarships and off scholarships. So students who really couldn't afford to attend universities or can actually attend on the university with the scholarship, sometimes even full parts of it is full scholarship, but some of them are part scholarships. And we also have a parity on gender in all our departments. We turned out to be one of the top 10 universities in Ghana and across the continent. And one of the mission of the university is to train ethical leaders. So ethical concepts are being really infused in our curriculum in all the different majors. And now we are expanding, doing all additional programs in the undergraduate. As I just mentioned, we just got approval for our biological engineering program, which would be probably the first of this sort of structure in West Africa. We are really looking at using, as the 3 highlights here, digital health, synthetic and computational biology, and neural engineering. Those are the 3 pillars, and we're trying to use bioengineering as being a bridge between our engineering, our engineering majors, the electrical, mechanical, and computer, and also computer science and business administration. And we're really putting in an emphasis a lot on entrepreneurship, hoping to have students starting their own ventures. We're also part of a large collaborative project with Canada on the health sector. And then we also have graduate programs. The MSc Mechatronics Engineering has been on for quite a while, I think it's in the 3rd year right now, or 4th year. And then we're starting 2 new master's programs. So if you'd like to come and join us to get an education, or you'd like to come and join us teaching even for a semester, or sometimes guest lectures, even remote, it would be great. Please let me know. We'll be more than happy to have you joining our classrooms and our communities as well. So I thought I'll present a bit of the work that has been done mostly by Rosamond, who's actually on the call, and she's been involved with a lot of the reclone work. And the idea was to rethink or re-engineer the cold chain. So this was work that we've done in collaboration and mentorship with Jenny Malloy, and she's been really mentoring Rosemont in collaborations with me through this work. So as we all are aware, the cold chain has issues in terms of high cost, expensive reagents. It is complex in terms of customs and regulations. I think people in the global south experience that quite a lot, and perhaps they're not people in the global north are not really aware of some of those issues. There's transport delays, there's transportation issues if they get and they're not stored properly, and then you lose all the purchase that you have made. There's challenges with storage, cold storage in particular, problems with power that could create issues, space inefficiencies. So these all create issues that limit the access of molecular biological tools all across the world. And so really the point that, or the protocol that we wanted to start with was really to start with cells that we can dry up in using very low resources, such as the vacuum sealing drying approach here, and then store them at room temperature and really analyze and understand if the cells are viable or and also if they are functional upon revival from this drying up. So we started with, so the reviving, so this was this is the storage protocol. So we isolated cells, we culture them overnight, we subculture them until we had an OD of 0.7 to 0.8. Then we harvest the cells by centrifugations. We aliquot them into the small PCR tubes, put them on the bed of the silica gel, and with the vacuum pump, we actually vacuumed, sealed the container, and allowed them to dry over 24 hours, after which we covered the tubes and we kept them at room temperature. And then we kept taking aliquots and checked for their viability and functionality. The reviving protocol was involved an initial rehydration of the cells, then which was in distilled water initially for 15 minutes. And we'll talk about the drying media in just a minute. Then we added media rehydration, followed by an incubation again up to 15 minutes, then resuspensions and the plating on the LB agar for overnight, and then the following day, looking at the colony and quantifying the presence of the colonies. We've assessed, we looked at an experimental design that dealt with cell viability and cell competency as a measure of functionality of the cells. We tried two different cell lines. This is the protocol for one of the cell lines, the BL21. Although we observed similar results in both of the cell lines that we worked with. So here's an example in what happened with the cells. So after drying, this is day one, the day after the drying, and then we continued this at different days up to day 50. So there is a significant decrease in the number of cells that you are getting the longer that you are keeping them at room temperature. However, really the point is that as long as you're getting some viable cells, then you can just revive them and continue on growing. And so we're going to look at the functionality in a minute to ensure that, yes, although they are viable, are they still functional? Can you still use them for functional assays such as transformations? We also noticed that we had a bit of variations between the different runs that we are doing. Hence, that's also shown by the large standard deviation that we have. and here's are just a couple of examples that shows that around day 160, we still had quite a bit of cells, but in the different runs, we would get different results. And those results were also repeated by our collaborator, by Jenny's lab as well. And I'll show you some of those results. Then we also compared them with cells that were stored at -80, which is in at least in this parts of the world, we tend to store them at -80 rather than liquid nitrogen. And so you can see that when we revive those cells, I don't know if you can really see, but it's basically a lawn of cells in this case, where in the ones that were dried up, you can actually see the individual colonies. So it's not, we have a significant reduction in the number of cells. But as we mentioned, as long as you have a couple of colonies, you're probably good to go and be able to use those cells. Then, as I mentioned before, we wanted to see if the cells are functional. So we used, as a marker for functionality, we looked at the competency. So basically, after we dried up the cells, we revived the cells, we inoculating a colony, then we transformed with DNA. We observed with a plasmid DNA, then we observed the results of the transformations, and we compared with the controls. So in this image here, we're looking at day 78, where these cells were transformed with PJump, and we use this particular plasmid because it gives fluorescence, so it's really easy to assess the transformation. And then we compare that with a control, which were cells that were -80 revived, went through the exact same protocol of transformation, the same concentration of plasmid DNA, and we had very similar concentration numbers. So even though we had this drastic change or drop in viability, as long as you have even couple of colonies, you can revive them, grow the cells back up, and be able to start doing transformations and be able to express proteins if that is the point of the experiment. As I said, this was done in collaborations and mentorship with Jelly Malloy. We sent some of those dried up cells to their lab to see if actually they obtain similar results. And so although they had a bit of less colonies than we did, we observed the same things, that we have cells that are being they're being viable and growing after the cells being dried off and transported to their lab in the UK, and then they have performed the same experiment. And they also did transformations for the functionality as well, and you can see that we're having again similar results. So this was a day 29, and this one's a day 297. So it's almost 300 days later, you're still having functional cells. And the other thing, this is with the BL21DE3. So that was the second cell line that we used. So we used both of these cell lines, and we saw similar results. They also seen the same things in between replicates. They have seen that they have quite a bit of variation. So it was not in a sense reproducible to ours, right? Because nobody really wants to reproduce that variability, but nonetheless, it appears that there is some variability between the different samples, and perhaps a lot more than we would normally expect. So really, to put it all into perspective, we have seen that the BL21 cells remain viable and competent after drying, that even at 78 days and even actually 300 days, they were still showing transformation efficiency. We saw that we could send the cells across the world and have another lab basically reproduce the same results that we have. And so I think the significance of these sorts of work is that now makes it a whole lot easier to send cells across the world. And in just a minute, we'll look at some of those cells that are even transformed perhaps with particular plasmids that we would want to send in case the labs to which we're sending them, they don't have the capabilities and the capacity to do transformations. You can actually give them functional bacteria that they are desiccated, and then they can grow them up and start producing enzymes. And so really, we hope that we convinced you that we were able to dry the cells in a very frugal way or convenient way, that we might say, that doesn't require a lot of capacity and equipment, and they can be shipped across the world and still have functional cells. So helping with the transport, helping with having these cells available for educational and research applications, and the conveniency of being able to transport them across the world. And then I thought I'll share a little bit of some of our other work that we are looking at. So as it was mentioned in the beginning, we are part of the Reclone community and serving as the African Center for the DNA collections. So we've been working with, these are preliminary results with using the open vent enzyme. So we use the open vent enzyme with the M-cherry and without M-cherry. And so we've been able to produce them both repeatably several times. We have been able to use them to be able to show that they're able to be used directly into PCR reactions. So this was the product that we were expecting. We can see that the ones in the case of the open vent plus the M-cherry, it has less efficiency, so you would have less product in this case. And we really think about being able to send, for instance, both of those to labs, because it makes it quite easy to assess if your transformation and your protein production is working, having the M-cherry. And then you can do them side by side and hope that, right, the one without the M-cherry still behaves the same way. And here we can see that they're both functional. We are currently working to compare this with current enzymes with the Taq polymerase, and so those experiments are underway. And then the part here on the bottom is that what I was trying to explain before is that we took the dried up cells and we transformed them with open vent M-cherries. I think this was early into, so apologize for that. It's not one of the biggest the best image in terms of oh, actually, no, never mind. Sorry. These were not induced, so they were just transformed. So we are checking, and we do have some, unfortunately, I don't have that picture where we can see that we've also had induction as well from these particular cells. So the ones that we have here were not from dried up cells. They were from cells in the -80. So these were the ones that were dried up before. So the idea would be that if anybody would like to have this or receive this enzyme and they don't have the capability of doing the transformation, we can send them the dried up cells that have been transformed with the enzyme, and all they have to do is go through the protocol of reviving the cells and then start their protein production. We have worked on the venture, so we have the beginning of a venture. We are a bit in the ideation stage, trying to develop the viable product, sort of, where we are trying to create this venture on campus where we can start producing a lot more of these enzymes that come from the Reclone repository. We don't have yet the second part of here, so we would like to acquire and start being able to do some purification as well, more or less for research purposes, right? So for demonstrations and teaching, I think this is not really necessary. We can send them the cells and they can just start producing them without having to have the actual enzyme. But if you would like to, we would like to be able to sell enzymes produced on the continent and locally. And for that case, I think we would like to be able to purify them and be able to provide pure enzymes and be able to quantify the activity of the enzymes and be able to provide high quality products to research lab and diagnostic labs locally in Ghana and or across the continent. So we're making progress towards this. We're not there yet, but we are making progress, and we hope that fairly soon we'll be able to start producing high quality enzymes that we could share. And then a little bit more on, I guess, other projects that we've been working on. And this is more kind of a pitch to ask for people who are interested to collaborate, to work with. And so we've been very interested in trying to work with hydrogels as an improvement towards sustainable agriculture, first for irrigation purposes, and then also for delivery or incorporations of agro bacteria for fertilizers, for natural fertilizers. So what we're showing you here is that we were able to create hydrogels out of cassava starch. So we basically did the extractions of the cassava starch on campus in our labs and compared them with PVA, for instance, which is one of the traditional hydrogels that have been used in agriculture. Then we've been able to show that you can use the cassava starch to actually grow plants. So these are bean sprouts that have been either watered or just grown in the presence of the hydrogels. So we're able to release that water and allow the plants to grow. And then below here, I'm just showing you that in the same in hydrogels, although these particular hydrogels are made out of alginate, but we have shown that the starch actually behaves the same way, has the same mechanical properties as the alginate. It's prepared in the same way as the alginate is, and then it's cross-linked the same way with a calcium chloride. So we're in the process of incorporating bacteria into the cassava starch hydrogels as well, but these are from alginate. And what this one shows that we were able to incorporate two different cells. These were kind of green fluorescent proteins, and these are red, and then this is just the alginate alone. And we've been able to show that they can stay viable within the hydrogels for up to 5 days. We just didn't do any longer than that, but we're working on extending that period of time as well. And we also can show that we can incorporate different numbers of cells, quite a bit of number of cells, up to 15 million cells per bead of hydrogel. And so the point is that we wanted to be able to also calibrate or be able to have a calibration course also for delivery of the fertilizer in that sense, if we wanted to, through the different number of cells. And then this is another project that we've been working on. This is a final year project where, in collaborations with a research lab from Kenya, we were interested in trying to find a way in which we can design a test for tuberculosis. It started being motivated by tuberculosis in TB patients, which have a low number of bacteria in their sputum, and it's also very difficult for them to produce the right amount of sputum, which is quite significant. And also by doing sputum cultures, those take up to sometimes 3 to 4 weeks, which is problematic in terms of treatment for HIV patients. So in this particular case, we had combined microfluidics with LFA on the side. So the idea is that the sputum is being loaded in this chamber, and then we have some of the addition here in this chamber. We can add sodium hydroxide, 8% sodium hydroxide, and we can even alter a bit of the pH. So it seems like if we change a little bit the pH and bring it up a bit more towards neutral, we actually are able to dissolve or thin out the sputum significantly, that it can actually we can actually collect the bacteria from the sputum. Now, we didn't work with real sputum. We worked with a synthetic PVA at a fairly low concentration, has been used as a analog for sputum. So we basically created PVA gels and we added the fluorescent bacteria in it, and then we've loaded them in here. In addition, we also added a motor, so we can able to spin the plate, the microfluidics here, and that allows us to basically put whatever sputum hasn't really dissolved to settle at the bottom of this chamber, and then be able to just allow it to flow just the bacteria through the channel into the second chamber. And then into the second chamber is where we are looking to do cell lysis and be able to transfer the cell lysis then into the LFA and be able to test for the presence of mycobacterium. And in this case, we were investigating using aptamers rather than antibodies on the LFA. We haven't been able to test that. We have the aptamers from the literature. It wasn't that we did that research, but we just haven't received all the parts in time to be able to show the LFA working. What we have been able to show is that in this particular case here, you can still see some of the sputum is really quite still thick, and so upon spinning, it kind of aggregates at the end, at the edge of the chamber. But then you can see that some of it's actually making it through, and we're able to see it on the LFA architecture. So this case is this is no DNA, it's just the bacteria. And then we're having some big problems with gluing at this point. So we're looking at alternatives of being able to get some better assembly here between the layers of the microfluidics. So this work really was inspired by our collaborations with the Global Frugal Diagnostic Network with Maywen, who's here, and we started from that collaborations and the workshops that we've done on campus, and we'd be able to get students to actually work on this. And I think I'll leave it here, and giving my thanks to the Global Frugal Diagnostic Network, to the Reclone, to the students who've been working on Rosemont and Martha and Prince, who actually helped us get things started as well in the lab. And I like to just put it into perspective that the students who have been working on these projects, they're undergraduate students with in an engineering field. So Rosemont started as an electrical engineering, and now she's been doing a lot of the iGEM work, and so she has gained a lot of knowledge in biology. And Martha, who is a mechanical engineering student, and she also worked with iGEM, so she's gained a lot of biology knowledge, and she's quite interested in this sort of work. So I think I'll stop there and take questions.
Amazing. Thank you. Thank you so much for sharing. Truly amazing, inspiring work. As always, I'm very impressed of how many things our community does and how our community has been really moving and pushing forward the work in biotech, open science within your countries, their regions. Thank you so much for sharing about the journey, the projects, really, really exciting things. And yes, now we can open to questions, comments. I already saw potential, even more collaborations happening on the chat. Oh, yes, please go ahead, Atef.
Hello.
Yes.
This is Atif from Egypt. I would like to ask about the transformation. When you said you did transformation, Elena. Thank you for your talk. It's very impressive, by the way. I like your idea a lot, because I'm suffering also from the cold chain here in Egypt, because it's not in my university. I came from Saudi Arabia, stuck in Egypt for some while. So for the cold chain, I'm suffering from it. So very, very interesting for me. But when you say you're doing transformation or making content and doing desiccation or drying by tree laws or your protocol, this is the chemical competent cells or electrical competent cells?
No, chemical competent cells. And we're making them competent after the revival from the drying procedure.
After, okay,
Yes.
that's after, okay.
Yes, yes.
So you have to do very, very small batch because sometimes you are doing 1 or 2 transformation batch. Okay, thank you.
Yeah, yes. Eric, please go ahead.
Yeah, that was a great talk, and this is something that we think about a lot at Adgene, and I am wondering, we've got around sending transformed bacteria by just sending bacterial stabs, basically, and at room temperature or at ambient temperature, and which does have some risk because, if particularly in hot regions, those can die during transport. But I'm just wondering if that was something that you've considered for some of these things, is just sending it without the desiccation process, because that does add, I guess there's advantages and disadvantages to both, but it is cheaper and faster for us to send things just as bacterial stabs. So I'm just wondering if that's something that you've thought about at all for some of this.
So I think we did, but the point was that we knew that that's something that's common practice, right? So we were looking for an alternative to that, first of all. And also, I think it's a bit, also shipping from here to somewhere else, it's a little bit more difficult. So I think going in with a stab where the actual cells are alive, it's a little bit, they can see it because the other cells are alive too, even though they're dried, but at least they look like a powder, so they don't envision that as being at the risk.
That's definitely true. That's definitely true, that even though they're biologically the same thing and alive, just having it, being able to put it into an envelope or something like that is a lot different than putting it into a box.
Exactly.
Okay.
Yeah.
Thank you.
Yeah. So it's a perception issue, I think.
Yes, for sure.
Yes, Pablo.
Hi, everyone. This is Pablo from Argentina. Hope you are doing well. I have a very basic question. If I am not wrong, you rehydrate the dried cells with just water in the first place. I wonder if this doesn't produce like an osmotic jog and you are losing more bacteria than you would lose rehydrating them with media directly. I'm okay with. I agree that as long as you have a couple of living bacteria, you're good to go. But in some cases, you need the best viability possible.
Agreed. We actually tried before with PBS, and PBS alone, we lost a lot of cells. And then we also tried PBS and Trehalose together, and we also lost a lot more in that case as well. I don't know, Rosamond, you're on the call. I don't know if we went directly to media from Trehalose. Rosamond, do you remember? No, she's on the call, but maybe she can't talk. I don't know. Or maybe she's not anymore. We might have lost her. So I can't remember if we actually did try directly to the media or not. Oh, yes. So she'll respond. She says it's on the road, so she couldn't speak. But maybe she'll tell us if she tried. I can't really remember.
So you have better results with water instead of PBS or PBS with Trehalose.
Yes. So PBS
do you
alone,
have
that's
any?
What we tried. We tried with PBS alone first.
Do you have any clue why that happened?
We think that the Trehalose creates crystallization around, based on the literature that we've seen, that creates a crystallization around the cells, which protects them in the drying and through the revival process. That's based on literature. We haven't. Unfortunately, we don't have the microscopy and that capacity building to be able to do that. So, but based on the literature, that's kind of the guess that we're having.
Okay. Thanks. Thank you very much.
Sure. Maywen, please.
I just wanted to mention that as always, I wanted to invite everyone to anytime you have also exactly this troubleshooting questions. We have our forum, and I've seen a lot of amazing discussions exactly on, oh, I'm having trouble with this, or oh, I'm trying to do this. What have you done? What has worked? What hasn't? Just wanted to remind everyone about our forum. And yes, please, may we ask your question?
Elena, it's fantastic to see an outcome of the Global Frugal Diagnostic Network being new foray into microfluidics. Really, really pleased to see that. So I was wondering how you were, like, if you can walk me through what prototyping you used. I guess you laser cut part of the device. Yes. I'm interested to know how you found sourcing the acrylic sheets, whether that was easy. And then I'm interested to know, the bonding approach you took and what, maybe I can help there. Yeah.
Yeah, so with the acrylic sheets was not a problem. Those we had, and we had some left over from you as well, so we were fine there. The 3M bonding is the problem that we've been having. So we've been trying super glue, and then the super glue creates these air bubbles, and then you're having problems with leakage, and it dries up too fast, so it's really hard to assemble and align everything. And so that was the biggest problem that we've had. And also, if you're doing the fluorescence work, it's all fluorescence underneath at the same time. So you always kind of have to have two, because you're having really high background fluorescence from the glue itself. Okay. So we're still working on that.
Just from looking at your slide and the dimension that's, were involved there, I would think that the 3M double-sided adhesive should work really well. So I'm just wondering what sort of problem you have there.
We didn't have any, so we don't have any.
Okay.
No, we didn't have any. Yes.
So
I couldn't find it here.
I can buy a roll of these for you and ship that, because it's by far the easiest solution. It's not.
Agreed.
It's not completely foolproof. You have to adjust the laser cutting parameters so you don't burn too much of the tape. Then, of course, there's the alignment as well. But it's really good for when you start to prototype something. And of course, sometimes it can interact with what you have in the channel. There are tapes that will be more or less susceptible to these kind of things. But again, like a standard 3M double-sided adhesive, at least it enables you to demonstrate the fluidic function of your device really well. But then what you could, if you want to continue in that direction, you could build yourself a little bonding platform as we have in my lab, which is a low-cost just press. It's made for jewelry, and it's kind of $200 or so. And then you'd have to work with your sort of mechanical workshop for somebody to do a little heated block, but that's like really basic. I can share what we have also, and yeah, you could replicate, because then you can just use a little bit of ethanol, and that reduces the glass transition temperature of the acrylic. You press, you don't need a lot of tonnage or anything, and and then the nice thing is that you have like almost zero residues. You should have really good bonding and no problem in terms of biocompatibility.
Thank you. So yes, we'll follow up on that. I think I've tried getting 3M and couldn't get it anywhere. So we've tried other stuff.
Yes.
Thanks. Yes, Prince.
Can you hear me?
Yes.
Okay,
yes,
right.
You can.
Okay. So on your slide, you talked about when you were trying to measure the enzyme activity of the protein you expressed, you kind of made a conclusion that the activity was lower compared to the control you used, which probably is a commercial enzyme. Did you calculate the unit unit activity of the enzyme to compare to how much you're using from the commercial enzyme to make sure, like, to have that same unit comparison?
No, so unfortunately, I think maybe I misspoke and I wasn't clear enough. The comparison wasn't between the enzyme expressions and the commercial enzymes. We're working on that one. We haven't, the slide that I showed was between the open vent with M-cherry and without M-cherry. So that was the only comparisons between them. So the one without M-cherry had a higher activity than the one with M-cherry. The other ones we have been working on it. Unfortunately, we had some problems with our -80 with the power, so we kind of lost all our enzymes again. So we're in the process of reacquiring new ones, and so we haven't been able. So we've tried with we've tried a couple of several times, hoping that the enzymes were okay, and we just haven't gotten any product basically with the commercial enzymes, but we got product with the expressed enzyme. So that's a good indication, right? That this is a good thing to have. So we're working on getting new enzymes and then compare and calculate enzyme activity as well.
Okay, thank you.
Thanks. So I think Rosamond had answered that we tried with the media, but I don't think she understood the question. So when we dried up the cells directly in the media, then we lost the viability after a few days. But Rosamond, the question was if we've tried to resuspend the one that we dried them in trehalose, to resuspend them directly into media and not the water. That was the question. So if you can still hear that.
Sorry.
Did we?
Yeah, we did try that before. The activity was, the viability was very low.
Okay. Yeah. So that's the answer. That I think that's why we had to do the water first incubation and then adding the media and then allowing it to rehydrate before we plated. So thanks for reminding me of that. Yes, Maywen.
Yeah, I'm just thinking you could also 3D print the device, then you don't have a bonding issue. If your dimensions are above a cross section of the microchannel being above a cross section of 1 by 1, maybe just you could go 2 by 2, then because you also don't have a complex network, it'd be easy enough to clean inside your channels. And then you'd be sorted. Again, you might also have
but you'll clean with what? With acetone?
Yeah.
Okay.
Yes, with the normal solvent that you use for cleaning the
okay.
Or isopropanol, I think is what the SLA platform or any parts produced with an SLA platform you clean with 2 baths of isopropanol. And then you might just solve nicely.
I didn't think about that.
Your problem like this.
Yeah, that's really cool. Thank you. We'll definitely give it a try.
Yeah, and I see Atef is asking about the bonding approach that we've taken when we use the layer by layer prototyping. So I'm just going to type this in the chat quickly.
Thank you. And then maybe we can take them and even put them on the Reclone forum as well, so people can have access to them if we don't mind. I think.
No problem.
That's amazing. Well, do we have any other questions, comments, more ideas or collaborations? Yes, please go ahead.
Sorry. So you mentioned that the one with M-cherry has a lower activity than one without M-cherry, right? So you think the M-cherry is interfering with the activity of right?
I would think so.
I think Reclone designs Open Bioeconomy Lab designs their system to have a clean side. So if you could try cleaving it, I think we should get back to having a higher activity compared to post amplification. You should have if you use a cleavage enzyme, you should be able to cleave it off and have pretty comparable activity like without the insert.
Yeah, cool thought. So we'll do that in the next, as we're doing all the comparisons between them. We'll definitely try to do that as well. So that's a really cool point.
Amazing.
Any more questions?
Yes, and
Go ahead, please.
yes, I saw a lot of interactions on the chat. So also, if you're interested, please feel free to send your questions over in the chat. Also, as mentioned many times, we have also our forum to continue discussions. Okay, so it seems we have all questions covered for the moment. I want to take again some time to thank you, Elena. Thank all of your team at the Ashesi and all other collaborators that have been really helping us grow Reclone and our work and our reach in Africa. And thank you so much for everything you've been doing. I had contact with Rosemond and everyone also from the iGEM team, and it's really amazing to see the impact and how you have now people all over the world. So thank you so much for all this inspiring work, and thank you everyone that joined us today. Hope to see you all on our next community call in May. And yes, thank you.
Thank you all, and thank you so much for the opportunity, and great to connect with everybody again, and looking forward to continue on working together. So thank you so much.